Okay, good evening, everybody. I hope I'm audible. Today is session 7 out of 12, and we'll be starting in a couple of minutes. Let's wait for everybody to join in. I hope the annotations are visible as well as I'm audible. If it is okay, fine. Okay, good evening. Welcome to week 7 of the live interactive session of the course Genome Editing and Engineering from NPTEL. This course is taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket Mukherjee, a Prime Minister's Research Fellow from the School of Biological Sciences, IIT Delhi. Today's week is very important. It deals with CRISPR Cas9, which is one of the um, latest tool of genome editing in this field. So if you look at the discovery of this discovery of CRISPR, you would find out that in 1987, uh, there was a group of Ishino. They discovered, while discovering, alkaline phosphatase enzyme in E. coli, they found that on the 3' end, there were some palindromic repeats. And this is very important because these repeats were as such not important for the study, but they reported it. As they saw, they reported it. In 1992, Francisco Mojica, he got very interested in this repeats. So he was reviewing the genomic sequences from Halopharax mediterranea, and he found that there are 14 unusual sequences which were 30 base pair long, and they roughly read the same backward and forward, right? That is important. So another thing was that they were roughly palindromic, right? And they repeated every 35 base, so they were repeats, right? And Francisco Mojica was the person who coined the term CRISPR, which stands for clustered, regularly interspaced, short palindromic repeats. Fine. In 2002, Janssen, he also identified another set of sequences which are, uh, you know, um, are in conjunction or they work together with CRISPR, and they were Cas genes. CRISPR associated genes were identified. Now, these repeats, while they were very interesting to find in bacteria, in prokaryotes, however, scientists usually dismissed this, finding, because they thought that there are many repeats in a genome and everything does not make sense. So this entire finding was not very well accepted in the scientific community. It was kind of ridiculed also. In 2005, Alexander Bolotin, he found some spacer sequences. Okay, spacer sequence within the CRISPR. They were similar to the DNA of certain viruses, basically the bacteriophages that infect the bacteria. So to their DNA, these spacer sequences were similar too. And he also found 2 to 6 base pair protospacer adjacent motif. And that's why the hypothesis came that this CRISPR system is essentially a kind of adaptive immunity in bacteria. So that is how this finding became very, very important, right? So what happens next is, if we look at the timeline in 2012, there was the first in vitro gene editing using CRISPR, right? In 2013, we see that eukaryotic cells were modified. Eukaryotic cells were modified. In 2014, the precise method of gene editing was figured out with the help of CRISPR-Cas9 system. In 2016, the oncogenic mutation were inactivated using CRISPR, right? In 2017, so beta globin gene, which is very important for beta thalassemia, you know that. So that gene was modified in a human embryo using CRISPR-Cas9. So in 2019, Jian Kui, he was arrested for violating medical regulations for interfering with the human genome as such. So he was arrested because he used again CRISPR for this. In 2020, pretty recently, Emmanuel Charpentier and Jennifer Doudna, two female scientists, they were awarded with the Nobel Prize for discovering a novel method for genome editing. So in 2007, essentially, if we look at it, 2007, Barango et al., Barango and its group, they found the role of CRISPR in specific. So they found that CRISPR is important for adaptive immunity in bacteria against uh, phage infections. This was found in 2007. So essentially, the role of CRISPR-Cas in the bacterial system was figured out, and the methodology was so simple that we use this even till date to edit genes, right? And we'll see how that happens. So if we look at the CRISPR-Cas system, how is the locus arranged? So initially, there is a Cas operon. there's a Cas operon, and it is, essentially there. It is transcribed and translated to form Cas set of proteins. These proteins are very important for the cleavage function of CRISPR, right? But the recognition is actually done by the CRISPR array, which is again arranged something like this. And within this array, there are some identical repeat sequences. So these are some identical repeat sequences. And in between these spacers were similar to the phage DNA, bacteriophage DNA. So this CRISPR array was essential for recognizing the specific, DNA sequence, and the Cas proteins, whatever is translated after this, the Cas proteins like Cas9 or Cas12, we'll study about it. They were important for cleavage, right? That was how it how this system worked in bacteria. So upon phage infection, a sequence of invading DNA or protospacer is incorporated into the CRISPR array using two very important proteins, which is present in all the CRISPR systems. They are Cas1 and Cas2. These two proteins are common to all CRISPR systems, whatever types of CRISPR systems you will study. They all have Cas1 and Cas2. And the role of Cas1 and Cas2 is to integrate these, phage sequences or foreign DNA into the CRISPR locus, right? That is how it works. So if we look at the CRISPR-Cas system, based on the structure and function of Cas protein, can be differentiated into or classified into two classes, class 1 and class 2, right? Among class 1 is type 1, 3, and 4. And among class 2 is type 2, 5, and 6. 1, 2, and 5, these three are important for recognition and cleavage. Okay, let's write it here. Type 1, 2, and 5 are important for recognition and cleavage. Type 6 is important for RNA editing, whereas type 3 is important for both editing in both DNA and RNA. All known CRISPR systems, as we already discussed, they all have Cas1 and Cas2 as common. Type 1, type 2, and type 3, they are important for maturation of CRRNA or CRISPR RNA. And it targets the foreign nucleic acids like DNA or RNA, right? Type 2 has the large endonuclease domain, which is around 1000 to 1600 amino acid long protein called Cas9, which is important for uh, editing. Now, what happens is that there is acquisition of foreign DNA, and that happens through The protospacer, protospacer, as we already said, it is the DNA from the phage. Protospacer is binding and selection. Then there is a 3' overhang cleavage. Then there is integration of this, cleaved DNA into the CRISPR locus. And then there is DNA synthesis and repair. Fine. Type 2, if we look at type 2, they are they require an unique accessory RNA, like which is called tracer RNA. Tracer RNA. This tracer RNA is basically transactivating CRISPR RNA. Cas9, it acts as a scaffold for maturation of CRISPR RNA, and it cleaves the double-stranded DNA targets. So Cas9 protein, it has a PAM interacting domain. PAM interacting domain means protospacer adjacent motif interacting domain, which is responsible for specificity to this PAM. And this Cas9 protein has two main domains, two main catalytic domains, which is HNH and RUVC, which is important for the nuclease activity of Cas9. So we are talking about Cas9 here, right? If we look at the steps for CRISPR immunity, what are the steps? The first step would be adaptation. Adaptation means where when a new set of DNA it gets inserted into a bacterial cell, it then it is inserted or it is integrated into the CRISPR locus. The second one is expression or biogenesis, where there is transcription of this CRISPR locus, which acts as the guide RNA or sgRNA also. Short guide RNA is what we know. It acts as a guide RNA. And then there is interference. So the CRISPR associated proteins, they come up, they detect the foreign DNA specific sequences based on this guide RNA, and they cleave it. Fine. So now is the interactive part of the session. So question number 1, the term clustered regulatory interspace palindromic repeats was coined by which of these 4 scientists? You may answer the question if you want. Okay, so the answer to this would be Francisco Mojica. So we have already read about CRISPR history, where while discovering the alkaline phosphatase gene in E. coli, there were several repeats which were found, right? And based on this, Francisco Mojica in 1992, he also found certain repeats, and he termed this very repeats as the CRISPR, clustered regulatory interface um, short palindromic repeats, right? But as I already said, that a lot of scientists, they ridiculed this work. They did not accept that these repeats would have any relevance. But anyway, so we know the history part very clearly. This is the entire timeline that we have already discussed. And then coming to question number 2, type 2 CRISPR-Cas system is defined by principle large endonuclease Cas9, which ranges in which of the following amino acids?
Option B, sir.
Option B.
Yes, sir.
In this case, it should be option D, 1000 to 1600 amino acids. So we have already talked about Cas9 system. It is part of type 2 CRISPR-Cas system. It has 1000 to 1600 amino acids. Okay. Which of the following is not a key step of CRISPR-Cas immunity? Is it adaptation, expression, interference, or reproduction?
Reproduction, sir.
You're right. So we have already said how CRISPR-Cas system works. It stores the DNA of the infecting virus in the CRISPR locus, right? And whenever this same bacteriophage will infect the virus again, since it's already there in the CRISPR locus, it's very easy for the bacteria to cleave this DNA. So it's basically a way of acquired immunity or adaptive immunity in archaeas and bacteria. And before CRISPR, it was not known that prokaryotes have this kind of a system very similar to adaptive immunity. Now, if you look at the adaptive immune system of humans or higher order organisms, it's very complex. And it also works principally in the same, I would say, same concept that any pathogen that infects you, your body can develop immunity against that pathogen. Even if it is a novel pathogen, your body can produce antibodies against those novel pathogens. In case of humans, it's a little bit more complex because it's not only DNA based, but also structure based. For example, if a virus infects you, the antibody will again go for recombination. As you all, if you have studied about immunology, you know about VDJ recombination, and they recognize these virus particles as foreign objects, and they essentially develop immunity. But anyway, the point is that this kind of a system was first identified in bacteria. So what are these steps we have already discussed? So adaptation and then expression and then interference. Reproduction is not at all related to CRISPR-Cas9. It has nothing to do with it. Rather, it's a system of immunity. So we have already discussed this, so we are not going any further. Neurodegenerative diseases are characterized by the progressive loss of neurons in dash and peripheral nervous system and the deposition of proteins with altered physiochemical properties. Is it heart, liver, brain, or kidney?
Brain.
Yeah, you're right. Brain would be the answer. So neurodegenerative diseases, they're characterized by the progressive loss of neurons in the brain and peripheral nervous system and the deposition of protein with altered physicochemical properties. If you look at the common neurodegenerative diseases like beta amyloid is these are aggregates of proteins, or alpha synuclein, which happens in Parkinson's disease, beta amyloid in case of, I think, Alzheimer's, Alzheimer's disease, Huntington protein, again, in case of Huntington's disease, prion protein, tau protein, all these proteins are there for specific, neurodegenerative diseases. Now, in case of the therapies that are involved for neurodegenerative disease, there could be the disease modifying therapy, where you have like platelet derived growth factors, glial cell line derived neurotrophic factors, or brain derived neurotrophic factors. These are several disease modifying targets that can decrease the development of, let's say, neurodegenerative disease like Parkinson's. There could be non-disease modifying therapy, where there are several factors which induce dopamine synthesis, because dopamine, which acts as a very important neurotransmitter, is non-functional. It doesn't work. In that case, that leads to the exacerbation of this disease, Parkinson's disease. And hence, factors which induce dopamine synthesis, they are, or their analogs, they can work as non-disease modifying therapy. So you can read about all of this in the slides. I'll not get much into it. Um Question number 5. Which of the following vector is known for high efficiency of delivery and is widely used for the delivery of CRISPR components inside the cell for gene therapy? Is it adeno-associated viral vector, RNP, DMD, or AADC? Any answers? Okay, so adeno-associated viral vectors in general is very much used in case of uh, high efficiency delivery in of CRISPR components, and not only CRISPR components, a lot of in other molecular biology and recombinant DNA approaches as well. When we have to insert a gene, adeno-associated viral vectors are used. So CRISPR components, like if you remember the entire locus as the entire construct of CRISPR, which includes the Cas proteins as well as the CRISPR locus. They are packaged as a plasmid DNA, and the challenges which are there associated with adeno-associated viral vectors is that it can cause viral toxicity, right? It could have some uh, it has a long expression because it has an infectious cycle. It can infect multiple generation, and what happens is after a few generations there are mutations which happen and that causes some off-target effects. So to avert these cons, there are other methods, right? So for ex vivo delivery, there is microinjection, electroporation. Then there is the delivery of Cas9 protein and the guide RNA as a ribonucleoprotein complex. That is also there. So as you can see in this slide, there are multiple CRISPR components which can be delivered through multiple methods into the cell. And the idea is when they will get into the cell, and this cell could be an eukaryotic cell, like in case of a human cell line, when they get inside it, it will be transcribed and translated, and they will function according to your needs. So because you can basically control the CRISPR RNA or the guide RNA, if this control you have, the Cas9 system remains the same, and if you change the guide RNA, then you can cleave any other sequence you want. So the point is there is a guide RNA library, which is very important for CRISPR Cas9 immunity, or I would say rather CRISPR Cas9 genome editing, right? Okay. Anything else from here? I don't think. Okay, there are ex vivo delivery approaches also, where what happens. So again, if there are some disease cells, those disease cells are taken out from the body. Like in case of biopsy, it can be, so let's say there's a tumor, you would take out those cells and then you would edit these cells. And once these cells are edited and you have the good cells, or you're mutated, what you have created the mutation what you want, then you insert these cells back into the body. So that's how ex vivo delivery works. In case of in vivo delivery, what happens is you directly infuse these components right into the cell, and these components are transcribed and translated in the cell, and then what happens is there only it does its work. So the problem with this is that all cells never undergo um, this gene editing. So there are few group of cells which will still remain, diseased. They will have the diseased phenotype. Few will, let's say, recover. So if you're working with a disease like, let's say, sickle cell anemia or even beta thalassemia to some extent, if it is the minor form of the disease, then probably in vivo delivery is possible, but otherwise it's not. However, as far as I know, CRISPR systems are not yet used in therapy. Uh, The design of single guide RNA is crucial to reduce the dash, as it is the sole guide to control Cas9. Is it Duchenne muscular dystrophy, off-target mutation, Cas9 variants in orthologs, or heterologous nucleases? Any answers?
B.
You're right. B is the answer. So the entire idea of the single guide RNA is to prevent off-target effects. So the designing this single guide RNA is very important. In the lectures, you would see various applications of CRISPR. There are several web tools of CRISPR, and for very known or the genes which are usually worked with, those genes, there are already guide RNAs which are there as libraries. They are already reported. Those also can be used. Okay, that's about single guide RNA. Question number 7, specificity of Cas9 nuclease cannot be further increased by choosing the target sites with few or no close relative in the genome, using truncated guide RNAs, employing paired Cas9 nickase instead of nucleases, or dissolving Cas9 FOC1 fusion proteins. Any answers? So specificity of CRISPR can be increased by choosing target site with few or no close relatives in the genome. It's very important, because if your target site is present or similar target sites, or if your target sequence is not unique, then CRISPR could cleave at off-target sites. That's obviously there. So if you choose a site which is very rare, then that problem is solved. Truncated guide RNAs can be used. Right? Paired Cas9 nicases instead of nucleases could also be used, and utilizing Cas9 FOC1 fusion protein. This is the answer. So dissolution will not be the answer. Option D is wrong. So yeah. Inteins or intervening proteins are in frame intervening polypeptides involved in DASH by an unique auto processing where it excises itself out and form of peptide bonds between two flanking external proteins. DNA splicing, protein splicing, RNA splicing, or enzyme splicing. So inteins are in frame intervening polypeptides involved in protein splicing by unique auto processing. So where it excises itself out and form of peptide bonds between two flanking exteins. That's how it works. Mutation in betaine aldehyde dehydrogenase leads to the production of aroma in rice, color in rice, bigger size in rice, or more carbohydrates in rice. These are there in the slides. This time the week has been a little bit heavy, but it's one of the most important weeks in this course. So mutation of this gene is used to produce aroma in rice, right? That is what is the answer. There are over 40 commonly used guide RNA designers which fall into one of the following three genres only. Find the option where they don't fall under. Is it pattern recognition genre, feature rule genre, machine learning genre, or position finding genre? So position finding genre is not there. So when guide RNAs are used, they either have, you know, they are based on how they recognize a specific pattern, right? That could be one of it. Another could be the features which are present. That could be one of the reasons how it recognizes a DNA of interest. There could be machine learning also, which involves certain algorithms which works behind this. So you can read about all of these in the slides. I have kept it. Question number 11, which of the following is not a web tool for genome editing? Is it KIME, ChopChop, CRISPR, or eCRISP?
Option B, sir.
ChopChop is actually a web tool for genome editing. So if you look at the last lecture slide, there you would find that these three are already present. Chyme is used for microbial next generation sequencing. That is how it's what it is used for. To analyze query sequences in NGS data, B analyzer doesn't require a full wild type sequence for reference. The type of base editor, the desired base editing window, or non-target DNA sequence. Non-target DNA sequence is not required. It actually requires a target DNA sequence. So users need to upload their pair-end sample NGS data. As an option, they can additionally upload data from a CRISPR untreated control to compare it with the data of the treated sample. So CRISPR treated and CRISPR untreated you can use. Next generation sequencing is a sequencing method to find out whether your whatever genome manipulation you wanted to do has actually happened or not. So to analyze query sequences in NGS data, B analyzer, it requires these four things. So full length wild type sequence that is used as a reference. A type of base editor is used, and then a base editing window is selected, and then there's a target DNA sequence which you want to see where if there has been any modifications in those sequences. Question number 13, which of the following is true about the function of Cas protein? Type 1, 2, and 5 systems recognize and cleave DNA. Type 6 recognize and cleave DNA. Type 3 can edit RNA. Type 2, 1, 2, and 5 systems can edit both DNA and RNA.
Option A, sir.
You are right. Question number 14. Which of the following is not a correct match about the signature protein of CRISPR-Cas system? Type 1 is Cas3, type 2 is Cas9, type 3 is Cas12a, or type 4 is Cas12a. Which one is not the correct match? This is again given in the slides. You can read it. CRISPR spelling is wrong. I'm really sorry. So type 3 Cas12a is the wrong option here. Which of the following is not a part of the 7 domains of Streptococcus pyogenes Cas9 protein? Is it Rec 1, 2, 3, bridge helix, RUVC domain, or Rec 4 domain?
Option C.
RUVC domain is a part of Cas9 protein. And it's so important because RUVC domain is important for the nuclease activity of Cas9. So as we already discussed, Cas9 has RUVC and HNS, which are the catalytic domain, which are important for nuclease activity. Rec1, Rec2, Rec3, and bridge helix are also a part of it. What happens during the adaptation step of CRISPR-Cas9 immunity? Transcription of CRISPR locus and processing of CRISPR RNA, detection and degradation of mobile genetic elements by CRISPR RNA and Cas protein, insertion of new spacers into the CRISPR locus, or transcription of CRISPR RNA. What happens during adaptation?
C.
You're right. CRISPR-Cas9 has been used to target Duchenne muscular dystrophy, one of the disease. What is this disease? A severe muscular degenerative disease caused by loss of function of mutation in the dystrophin gene located in the X chromosome. A severe muscular degenerative disease caused by the loss of function mutation in the dystrophin gene located on the Y chromosome. DMD is caused by exon loss, exon duplication, or disruption of protein reading frame in the 99 exons that compose the dystrophin gene. A severe muscular degenerative disease caused by loss of function of mutation in the dystrophin gene located both on X and Y chromosomes. What would be the answer?
Yes, sir.
You are right. Which of the following is not an example of epigenetic modifying enzyme? Is it histone demethylases, methyl transferases, histone acetyl transferases, or deacetyl methyl transferase? Deacetyl methyl transferase is no nothing like that. So that's the wrong option here. Cas9 mediated imaging allows visualization of dash in live cells to study the temporal and spatial behavior of dash in living cells. Is it chromatin and genome, genome and chromatin, enzyme and chromatin, or chromatin and enzymes?
A.
You are right. So Cas9 is basically the Cas9 is dead. It does not cleave the DNA. But this Cas9 has some fluorescent markers with it, okay, associated with it. So you can visualize the cell. So it will let you target the DNA. So that part will be there, recognition will be there, but cleavage will not be there. So you can now find out where in the you can visualize the chromatin in the cell, in the nucleus of a cell, and essentially you can find that where in the genome is that specific target sequence present. What nucleotide composition of an sgRNA, single guide RNA, is particularly essential to determine its specificity and efficiency? Is it the AT content, GC content, both of these contents, or it does not matter? Okay. Yeah. What would be the answer? No answers? It's the GC content which determines the specificity and efficiency, because GC content, GC have triple bonds, they are in that range. So the guide RNA in CRISPR-Cas9 is responsible for cutting the DNA, repairing the DNA, amplifying the DNA, or targeting the specific DNA sequence. This is easy.
D.
Option D.
You are right. What are the effects of U-rich seeds in the context of CCR5 gene editing using CRISPR-Cas? Is it sgRNA translation, sgRNA transcription, sgDNA transcription, or sgRNA mutation? Would be sgRNA transcription. You can read about it in these slides. So with this, I think that's the end of today's week's interactive course. If you, I would just stop the recording here. If you have any questions, feel free to ask.